So part 2, starting with 21. Remember that I recorded part 1. I extra recorded these questions. So part 2, what's an autotroph? They're organisms that make their own food, and examples of autotrophs are plants. They use the sun's energy to make food through a process called photosynthesis. And I'm going to go ahead and type that reaction right here. Give me 1 second. So plants make their food through a process called photosynthesis. And the photosynthesis reaction is as follows. 6 carbon dioxide plus 6 waters plus light gives you C6H12O6, which is glucose plus oxygen. So if you want to type the reaction by words, it's carbon dioxide plus water plus light. Those are the reactants. And then the products are glucose and oxygen. And let me move the paper over so you could see equal products. Okay, and that's the reactants and products for photosynthesis. You need to know that on the test. Now, when plants make their own food, if I didn't say this already because I lost track, heterotrophs eat this food, and this food is glucose right here, glucose. And then the oxygen is given off into the environment so we could breathe. ATP is made during cellular respiration, which is the energy molecule to run all cell process. The start of cellular respiration is glucose, which is the product of photosynthesis. Remember that cellular respiration is photosynthesis backwards. You just change the word light to ATP. So ATP is a molecule of stored energy. Glucose also is a molecule of stored energy because it's like potential energy. But ATP is for all cell functions. Glucose is for all living things. But glucose does aid in the process of making ATP. And ATP has 3 parts: adenine, ribose, and triphosphate. And remember that adenosine is adenine plus ribose. Okay, so that's where you get the name adenosine triphosphate. How does the energy in this stored molecule get released? Well, when you break a phosphate bond from ATP, ADP forms, energy is released, and a phosphate is made and is left to reform ATP. So as I mentioned before, cellular respiration makes ATP, and that is the process of making energy for cells. And here's the formula in written form, which is what you have to know on the test. Here's the photosynthesis reaction again, so you could see the inversions in chemical formulas. Remember that cellular respiration happens in 3 steps. You have to know the 3 steps, and you have to know where they take place. The first step of cellular respiration is glycolysis, and it happens in the cytoplasm. The second step, and if you want, add numbers here. So you remember this is 1, this is 2. The second step is Krebs, and it happens in the mitochondria, okay? And then the third step is ETC, and it happens in the mitochondria as well. All right, here are some diagrams you need to know. This is ATP. Remember that this is adenine, this is ribose, this is 3 phosphates. The energy is stored in the bonds. And when you break the bond, you get ADP, which will be missing this P, because it'll be adenine, ribose, 2 phosphates. Energy will be released, and there'll be a phosphate floating around. This is cellular respiration. Remember that cellular respiration happens in the mitochondria. The first step, so we would put this as a number 1, again, is glycolysis. The second step is Krebs, which is also called the citric acid cycle, but let's add the word Krebs there, because that's what you have to know. The third step is electron transport chain, okay? And in each of the steps, ATP is produced. You see it coming out here. Cellular respiration, you want to know that glycolysis starts with glucose. You don't have to know the in-between molecules. You have to know it happens in the mitochondria. Glycolysis happens in the cytoplasm, and it starts with glucose. The second step is Krebs, which makes ATP. Third step is electron transport chain, which makes ATP. They all make ATP. I also want you to know, if I go back here, because I forgot to mention this, that, over one more slide, that photosynthesis, if I could find it, right here, happens in chloroplast. So don't forget that. Photosynthesis happens in chloroplast. So after glycolysis, if no oxygen is present, anaerobic respiration will occur. This is also called fermentation. In humans, we call it lactic acid fermentation. And what that means is when no oxygen is present, your body will produce lactic acid instead of going through the 2 additional stages of cellular respiration. So Krebs and ETC will never happen. And that's that burning, aching feeling you get in your muscles when you're working out. Bacteria, on the other hand, will produce alcohol, and this is called alcoholic fermentation. So anaerobic respiration is no oxygen after glycolysis, and these 2 things will happen depending on the organism. In the presence of oxygen, we'll have aerobic respiration. So the main difference between these 2, obviously, is the presence of oxygen. So our next unit, which was cell division, or I should say the cell cycle, to be more specific, happens in 3 phases. 1 is interphase, 2 is mitosis or mitotic phase or M phase, and 3 is cytokinesis, okay? We're going to kind of go over each. So what you have to know, very, very important, is that interphase, there's no division. It's the prep phase. The division happens in mitosis and cytokinesis, okay? Mitosis divides cells, and you don't have to know this word, autosomal cells. We'll get back to that later. So let's scratch it out right here. You do want to know that mitosis has 4 phases: prophase, metaphase, anaphase, telophase, and cytokinesis is the splitting of the cytoplasm. You also want to know that interphase has 3 phases, so let's go over those because that's not here. Interphase. So interphase is when the cell's preparing for division. G1 is primary growth. S is the synthesis of DNA. And during G1, organelles replicate, and G2 is secondary growth. So once interphase finishes, then mitosis happens, and then cytokinesis. Remember that interphase is the longest part of the cell. Let me show you a picture of that. So you have interphase right here, longest part of the phase. It's represented here in blue. You got the G1, S, and G2. And again, here it is, growth, DNA replication, secondary growth. And then mitosis happens, and right after telophase is cytokinesis. So there should be like another little diagram here. So add the word right here, cytokinesis. Okay. Moving on. Mitosis, you have to know a little bit of details of each phase. So in mitosis, prophase is where you see the spindle, the centrioles move apart, the chromosomes become visible, and the nucleus disappears. You have to be able to recognize this and the order. Second is metaphase. You see the chromosomes down the middle, and the spindle attaches to the centromere. So you have to be able to label these parts too. So this is centriole, this is spindle, and these are chromosomes, and this is your nuclear envelope. There are your labels: spindle, centriole, chromosomes, nucleus. And then the last label is right here, this little dot. Let me make this a little smaller, but you put a little dot here, and that's your centromere, which holds the sister chromatids together, okay? And then an anaphase, so we got prophase, metaphase, anaphase. Remember PMAT. Anaphase is A for away, metaphase is M for middle. And then in telophase, you have the cleavage forming. You get the nucleus reappears, the centrioles move back together, spindle disappears, and the chromatins or the chromosomes lose visibility. And then you have cytokinesis, which is the splitting of the cytoplasm. Now, when you look here, I gave you a little overview of meiosis. We haven't really officially covered this yet, so we are going to scratch it off. But meiosis does have 2 parts, meiosis 1 and meiosis 2. You want to know that in meiosis 1, the chromosomes are cut in half. That's very important to know for the test. And then for meiosis 2, you want to know that it's similar to mitosis, The only difference is it starts with 2 cells because you have 2 cells being made here, and you end up with 4 daughter cells that are different from the original parent cell because they contain half of the chromosomes. So here's a little overview that kind of breaks up each. So mitosis happens in body cells or somatic cells. Again, you don't need to know the word autosomal yet. You know that mitosis has 2 daughter cells and they're identical to the parent. And the reason mitosis occurs is to grow, allow cells to, or people to grow, organisms to grow and replace dead cells. Meiosis happens in sex cells, which are called gametes, an example, sperm and egg. You end up with 4 daughter cells with half the number of chromosomes from the original parent cell. And this happens in preparation for reproduction. So, oh, here's the picture I ended up labeling. So you have to know your centromere, your centriole. Let's draw the lines to connect right here. Your spindles right here, and your sister chromatids are right here, okay? Actually, let me move that so it's not pointing to the middle, just any of that part. And then here's a breakdown of the parts of interphase, which obviously was out of order, but there they are for you to go back to and reference. Here is some genetics vocabulary for you that you have to be familiar with on the test. All of these questions are on the test or applied. Let's go over the Punnett Squares. So if I mention the word purebred cross, it's a homozygous cross or an F1 cross, and the offspring, so here are my parents, homozygous and homozygous, and he's crossing 2 different, so he could see what would happen. The offspring is called hybrid or F1 generation, and all the offspring are heterozygous. We're always going to use the capital letter of the dominant trait, so capital T tells us that tall is dominant to short, and we're going to cross a homozygous tall with a short, and these are my parents. The genotype of the offspring is capital T little t because genotype is always expressed with letters. These are a heterozygous. Phenotype is tall because if you're heterozygous for a trait, the dominant gene always masks the recessive. That's Mendel's first principle, which is called the principle of dominance. Then Mendel went a step further and he crossed the offspring. So he crossed the heterozygous, or he crossed what's called the hybrid, and that's called a heterozygous cross or a hybrid cross or an F2 cross. The offspring is F2 generation, and he ended up with these results. So the genotype results tell us that 1/4 of the offspring, every time you cross these 2 parents, can be capital T, capital T. Half of them can be capital T, little t, and 1/4 can be little t, little t. The phenotype is 3/4 tall because this offspring is tall, this one is tall, this one is tall. This one, on the other hand, is short, so 1/4 short. If I asked you what's the percent of homozygous, it would be 50%. This is homozygous, and 50% heterozygous. Let me go back to the vocab real quick. You don't have to know this slide. I want you to know that Mendel is the father of genetics, and he used Punnett Squares to